Welcome to the ASN Kidney Week podcast, exploring the latest advancements, research, and trends shaping the future of kidney care. Tune in for expert conversations, clinical insights, and highlights from ASN Kidney Week, a go-to source for staying informed and inspired.
Another exciting day at American Society of Nephrology Kidney Week. I am Vineeta Kumar, transplant nephrologist at University of Alabama in Birmingham.
And I'm Meg Jardine. I'm a trialist and nephrologist from Sydney, Australia. Really good to be here.
And hi, this is Bob Montgomery, and I'm a transplant surgeon at NYU Langone Health. I can begin by talking a little bit about a plenary session that I'll be involved in tomorrow. It's from 8 a.m. to 9 a.m. And it's going to involve 4 people, one of my colleagues, Leo, and myself will be talking about our clinical trials in xenotransplantation. So that's the main topic of the plenary session. I'll be sharing the current trial that we've just initiated, sponsored by United Therapeutics. And we'll probably talk a little bit about some of the single patient INDs that we've done as well. David Cooper is going to talk about sort of the history of transplantation in the xeno realm that, for the last 40 years has just involved putting gene-edited pig kidneys into primates, and it's only very recently that we've moved these organs into humans. So we're really looking forward to having a nice audience tomorrow. It's going to be a panel discussion, so we won't bore you with slides and formal talks, but really just kind of openly discuss what we think is ahead of us in xenotransplantation, toward making this the standard of care to try to reduce the tremendous gap that is getting wider every year in organ supply and demand. So that's what we're going to be talking about.
Yeah, so along with that landmark pioneering work in xenotransplantation with moving to inhuman experiences and now clinical trials, which you'll talk about tomorrow, it's also super exciting to have these landmark clinical trials in native kidney disease and native glomerular diseases that have the potential promise of extrapolating to post-transplant recurrence and glomerular diseases. So I was fascinated to hear about the atacicept experience in IgA nephropathy, in native kidney disease, and also have started to see abstracts trickling in with these other novel therapeutics between pegcetacoplan and iptacopan and C3GN. And so I'm excited about those abstracts that I've seen in this kidney week too. Meg, how about you?
Yes, it's been so many agents that are coming through, and you're right, the big standout has been the activity in GN. I think it's a tribute to the nephrology community. It was through the FDA and the nephrology community organising together to find pathways to get indications early enough that there was a rationale for developing. And we're seeing that 70 trials in IgA. It's just staggering. We wouldn't have seen it 10 years ago. So I find that really exciting, and as well on the backbone of all the development in the common alphanumeric diseases. The last 5 years has just been transformative.
And when you bring up FDA, the other corollary, and probably you could speak to it too, Dr. Montgomery, but borrowing from the native kidney experience of proteinuria and now talking about GFR slopes and adapting that to the transplant experience and how we can make those viable endpoints in post-transplant trials, proteinuria and eGFR slopes would be fantastic. And then, Dr. Montgomery, you've had experience with the FDA with xenotransplantation and defining post-xeno endpoints, if you want to speak to that.
Yeah, I mean, one of the things that we have sort of chronically suffered from in transplantation is that there really wasn't an acceptance of surrogate endpoints. And so the trials had to be designed and, with sort of the traditional transplant endpoints, which, essentially involved, the graft survival, allograft survival, and patient survival. And those kinds of trials take so long to complete, to finish it. Most of the big pharma groups that had been in transplantation kind of exited because it's just the drug development is so protracted because of the lack of acceptance of surrogate endpoints. But the FDA has definitely softened on this, and now is considering GFR, IBOX, a number of different new surrogate endpoints, and looking at 1 year endpoints and even 6 months in the case of xenotransplantation. So that, I think, is that plus AI being used now to inform drug development and search for new molecules, therapeutic molecules. I think we're going to see this tremendous explosion in new drugs that are going to be tested in the next 5 years. And we're going to see a lot more big wins like we're hearing about this week at this meeting. But, maybe we could talk a minute about the methodology and how to build trial platforms so that they're more nimble and we can get answers quicker when I think, we're going to see a lot more drugs that are looking promising that need to be tested.
Yeah, that's a really good point. We really have to make sure that we set things up so that they can be tested, or we'll lose the interest. Platforms are a great way to bring patients together. We've seen in oncology how efficient they can be to get patients into trials. I think just one of the big components there is you set things up for sites, so they can put their trial infrastructure in. They know they're always going to have a trial for certain conditions. We've got to adopt that approach in nephrology. And now that there are so many different agents being tested, it's the time to do it. The challenge with platforms is making sure that everyone has a stake, making sure that they're set up so that industry know that they will achieve their goals, that, I think there are hesitancies about any suggestion of head-to-head. I think that's overplayed. Many of the IgA trials that are being conducted now, they're essentially the same trial. So if people want to compare trials, they will. But in a platform, you usually don't compare head-to-head because everything's coming in at a different time. And in nephrology at the moment, the background standard of care is changing so quickly that you couldn't compare if you wanted to. So I do think that a platform for the glomerulonephritis in particular would be really efficient. And coming from a small population country, Australia, I'd really lobby for it to be multinational. I mean, that's the way to get the answers quickly.
Yeah, I mean, I've heard, members of the FDA say just as, exactly what you mentioned earlier, which is that transplantation, nephrology, we need to be more like cancer in terms of how we design, our trials and how we interact with our sponsors. One of the big things, I've been on a couple advisory boards lately with really interesting new drugs, and one other thing that I'm very involved in is the desensitization space. And we spend a group of thought leaders in that area spent two times recently the whole day designing a trial, and then we inquired about when we were going to start looking for patients, and they said, oh no, actually we're doing the phase 1, phase 2 trial in China. So this is a new thing too that I've been seeing more and more, and it has to do with the time it takes to begin to enroll patients in a trial. Now, the FDA will really insist on phase 3 trials being done in the US, but I think we're really missing out on a lot of important learning and research opportunities in the early phase trials if this becomes something that pharma does more and more. And so I think one of the things that we really have to learn from that is that time is money, and it also affects the back end of the patent. If it takes several years to get a trial through a phase 3, it's not just the money that costs, which is exorbitant, but the time is probably more important. So we have to get faster, be able to get these early phase trials through our institutions more quickly, through our IRBs and the contracting and all that. It's really a critical thing. Is that something that you've noticed in your experience? Because I know you're a really significant trialist, and just wondering if that resonates with you.
Oh, look, absolutely. I often feel that when you get the first patient in, half the job's done. The recruitment and follow-up, then it's just a matter of course. Not easy, but it's much more predictable. The unpredictable bit is all those things you talked about, the contracting, the regulatory approvals to get started. Australia at the moment has got the settings reasonably right. We have more people per million population in trials than any other country in the world at the moment. And that's because the settings are right at the macro level as well as the micro, having a stable health system. And I think it's a lesson there. It's not because there's anything different about how we conduct the trials in Australia, it's having the settings right. So I think that's super important. And you're absolutely right. You lose something if you're not part of the phase 1 and phase 2s, and the phase 3 is just brought in. The phase 2s are so influential on designing the phase 3. You do want them to be a little bit representative.
Right.
So jumping off of that point of phase 2s being so influential to design phase 3s, really exciting to see the BESTO trial, the tegoprubart, the CD40 IgG kappa ligand that was just released, the late-breaking trial. The safety signal looked really great on that. This is a drug that is being developed to minimize CNI use to reduce long-term metabolic side effects of calcineurin inhibitors. And in this randomized trial, at least the early phase data looked very good. Very minor leukopenia, some proteinuria in the treatment arm, but that was because there were 2 patients with recurrent FSGS in that, so that explains that. The rate of rejection was very comparable, maybe slightly higher in the tegoprubart, but nothing to worry about, much lower than with the Bella experience. The EGFR differences were not what they expected, but this wasn't really efficacy. It's a phase 2, so it's a safety trial. Some of the things to think about were they avoided patients with DCD, high KDPI above 85, and high cold ischemia time. Going back to your point that as they look to design phase 3, which they're going to look at efficacy, to include those very patients that CNI avoidance would actually give them the signal they're looking for would be absolutely brilliant. So I think it was exciting to see that they have a really good phase 2.
Yeah, I totally agree with you. I think this is going to be a really important drug in allo transplantation, because you can avoid the CNI effects on the kidneys. And also in non-renal transplantation too, because that's one of the biggest problems long term for heart transplants and lung transplants. And also, as we've been using more and more distressed organs, organs that from hypertensives, diabetics, older patients as donors, we're really looking more and more for alternatives to CNIs, because you can certainly see a really accelerated vasculopathy in some of those organs that already have injury to the microvasculature. and, you know, many of us have been using a lot of belatacept in those patients. So these, you know, what we used to call marginal organs. But that does come with, you know, issues with early rejection episodes. So the CD40 pathway is a really interesting pathway to interrupt, and I think it's going to be quite important moving forward. So hopefully, if we get good efficacy, it'll become available. Now it's been used sort of as a breakthrough drug in xenotransplantation in primates. And the question is, will that translate into humans as well? And now that we're starting human studies, you know, we'll probably get an answer pretty quickly to that. So the Mass General group working with the eugenicists is using the anti-CD40 ligand drug that you mentioned, and it complicates the trial, I think, a little bit, because you've got an experimental organ, the xenograft, the gene-edited xenograft, and you have non-approved drug that's, going through testing now. And some of the other drugs that have targeted that pathway have not made it through phase 3 trials for various reasons. We chose instead to use belatacept as the co-stimulatory blocker, which in xenotransplantation, it seems very important that you have a co-stimulatory blocking agent. So the trial that we're doing with United Therapeutics as the sponsors really involves only drugs that are approved, already by the FDA. But, we don't know the answer at this point, whether there's going to be benefits to using a CD4 monoclonal antibody. So we'll hopefully, by comparing these two trials, get some sense for that.
Absolutely.
Because the gene edits are pretty similar.
Your point about, the earlier point you also made about the extrarenal effects of CNI and what matters to patients is so incredibly important. We as clinicians focus on the lab and creatinine and eGFR, and our patients are also saying, I'm more than just my lab. I have a life, and that life does not want hair loss and tremors and down the lane, diabetes and all the other complications. So actually having alternatives and having interest in that field and developing that field is incredibly exciting.
Yeah, and there's a great, and maybe you can talk about it a bit, because surgeons, know about these studies, but they rarely actually read them. The study that was presented at a plenary session at the WTC about exactly what you're talking about, the side effects of immunosuppressive drugs, and a huge study that was done on transplant patients that I think was one of the most interesting things I've seen in a while, because it's sort of that voice that we have not heard much about.
Right. You're referring to the patient survey that was done,
Yeah.
US-wide, almost 10,000 patients that talked about life after transplant and what was the most distressing, and it was taking the medications and then living with taking the medication, the side effects of the medications, and fatigue, tremors, weakness. Those were the things that absolutely.
So tell me, are we getting any closer to a marker of biological immunosuppression?
Excellent question. Are we getting closer to a marker of biologic immunosuppression, sort of too much, too little, just right?
Well, then a lot of these side effects could be mitigated if we had a more
Yes.
precise measure.
Interestingly, there's actually been a lot of conversation about that. Lots of different biomarkers proposed, both in the blood, in the tissue, in the urine. And one particular one that's been talked about at this meeting is torptinovirus. So if there is a virus, for instance, BKRCMV, that's too much immunosuppression, and if rejection is too little, torptinovirus may be a virus that is just right. If you have a little bit of that, maybe that's just enough immunosuppression. We don't know that. It needs to be elucidated. So something as simple as that to, all the way up to profiling, immune profiling of different cells. The problem is it's a very complex field. When cells are recruited, not just it's not a binary system. Cells are recruited for a lot of different things. They're recruited to protect us, protect the organ, fight us. And so that milieu of defining what is the right phenotype for that particular patient is what we need to zero in. So the closest thing I know right now is the Star Trek tricorder. But
yeah, so this is a huge thing. Our immunosuppressive agents now are are much more specific than they used to be in terms of their activity. But we still really do not have, a good biomarker that tells us that we can reduce someone's immunosuppression. And most of the immunosuppressive reduction trials that have been done have just sort of randomly divided patients into control arm and a study arm and just tried to reduce their immunosuppression. Now we do have cell-free DNA now, which is an early indicator of trouble, and so that can guide immunosuppressive reduction studies, but I don't really think of it as a biomarker. It's more of like your ability to detect trouble, injury to the allograft early. So one that I think is really kind of interesting is eplet mismatch, and I think that is a promising biomarker. So and now in living donation, we can actually engineer low eplet mismatch transplants by doing exchanges. And this is being actively tested right now in a trial. The work from Canada, from the Winnipeg group, looks, as sort of the background information that is driving these trials that are being done. Looks very promising, as a biomarker for immune marker for being able to, stratify people into those that are likely to be able to undergo immunosuppression withdrawal or reduction and those that are not. So I think that's one of the most promising things out there right now.
Because it does seem, apart from the fact that you want to get it, we're trying clinically to get it just right for the patient. For drug development, if your biomarkers in effect is rejection that's dichotomous, you just lose so much information in that. If you could have some continuous, precise variable, I think there'd be so much entry into the market because there'd be promise.
Yeah, I mean it's just like the endpoints we were talking about earlier.
Yeah, exactly. Yeah.
We're just not that attractive to pharma, because of these things that we've been talking about. But I think that's going to change, particularly with the ease of being able to, using AI, select compounds and agents that, AI is going to tell us would be predicted to have, the effects that we're looking for. And as long as we can, set up good trials and get good throughput, I think we'll see a lot more progress. But one of the great dissatisfiers, I think, to our patients, and reflected in that study that we were talking about earlier, is really haven't been more than about one or two new drugs that have been added, to what we can make available for our patients in the past 20 or 30 years. So it's really kind of, for patients, that's been very disheartening, I think.
That's why the push now towards the surrogate markers for trial endpoints and patient-reported outcomes for trial endpoints, that will be huge, and that's what we're moving towards.
That's a great point.
Yeah. Would love to talk a little bit about rare diseases. We've talked about a few, like sort of touched on a few, but even rarer, I was really excited, especially since I recently had a patient with primary hyperoxaluria, PH1, and lumasiran. So they released their long-term results, so 5 years out, 60-month results, and so this primary hyperoxaluria type 1 is an autosomal recessive disorder with hepatic oxalate overproduction. And so very high serum oxalate levels, so that leads to obviously deposition in the kidney failure, and then when you transplant them, historically the isolated kidney transplant survival rates were extremely poor. So these patients, by reason, then needed liver, kidney at the same time. With the onset of this drug now, you can do isolated kidney transplant, leaving the liver for somebody else who needs the liver transplant. And then the drug keeps the oxalate levels in the serum low and thereby protects the kidney allograft. And the 6-month data is terrific. I'm sorry, this 5-year, 16-month data is terrific. The graft survival at 5 years is near identical to the control patients, historic control patients, without this disease. So I was very excited to see this, and I just convinced a patient that we should go ahead and transplant her rather than wait for a liver kidney, go ahead and transplant with the living donor kidney and use this drug. So very...
That's amazing. I mean, I can remember, early in my career, a lot of liver kidneys that we were doing for this disease, and a really devastating disease. So it's very exciting.
Sorry, Bob. Even just shortening the time to transplantation.
Yes.
With dialysis outcomes as they are, that alone could make a huge impact on whether the patient ever even gets to get a transplant.
Exactly, yeah. And the age at which they manifest is anywhere from 3 years on to adulthood.
Yeah.
So the really little kids get impacted by this all the way out.
And so in the trial, was the focus on transplantation or on primary disease?
I believe this is actually a phase 2, phase 3 study, and they just had the long-term follow-up results.
In native disease?
Yeah, but it was both native and transplant.
Okay.
Yeah, so they had, and then this was the survival analysis they did with long-term follow-up. And
that's been one of the wonderful things of the last 5 years, hasn't it? That
yes.
All these rare conditions. I mean, we used to not even be able to treat diabetic kidney disease very well, apart from ARBs and supportive measures. And now, not only are we addressing those very high volume conditions, we're also getting treatments for the rare disease, which are often in young patients, a lifelong burden.
Yeah, there's been a lot of investment in rare diseases. And, I think about sickle cell disease a lot, and the fact that we have a cure for it now, but it's somewhere, in between rare disease and, common diseases like diabetes. And so, the problem, we could eliminate sickle cell disease, but there's just not enough funding to do that because the interventions are so expensive. And so, it sort of sits in that zone in terms of its incidence, that makes it, I think, difficult to address unless we can bring down the cost of the treatment. And that's a shame because it has such a big impact on productivity, on people's lives from a very early time point. I think it would behoove us to really figure out a way to really address that disease and eliminate it because it could be done now.
This idea that it takes a village, listening to you both speak, the willingness of pharma to invest in our space, the willingness of our investigators to want to keep at it with a rare disease and follow these patients 5 years out, sort of makes me remember that I just saw these abstracts that are talking about the studies to come. So felzartamab, and looking at microvascular injury in kidney transplants, or the endothelin receptor antagonist farsantin, they're looking to reduce proteinuria. So there are some really interesting upcoming studies, which I believe at the future Kidney Week, hopefully we'll see some additional data from that. And that's another exciting thing to look forward to.
And maybe it goes back to what you were saying earlier about the borrowing from primary kidney disease into transplantation. We saw that happen at this conference with the borrowing that led to diabetes type 1.
Yes.
It's a fine study.
Yes.
And that is so encouraging. That's such an underserved population, and that younger people and have a huge burden. And the reason to think that the disease might be that different, that the evidence isn't applicable, that was challenging. But we need a mechanism, and now we have one. So I think for type 1 diabetes, for paediatric populations, for transplant, there's going to be so much that can come if we can get this coordination across our broader field.
And to hear transplantation being referred to as a rare disease blows my mind, because that's what I do every day in, day out. So I dream of the day where transplantation is not a rare disease.
Well, it's a rare disease only because of organ supply.
Yeah, that's
And if we can eliminate the wait list with xenotransplantation, with bioartificial organs, which are probably the next thing behind xenotransplantation, and get patients transplanted before they become disabled, which is common on the waiting list because it's so difficult to undergo dialysis and work, and then all the comorbidity that gets accelerated with dialysis. So we're transplanting patients, at a point many years later than we should be, and as a result, it's a much greater burden on their lives long term. And I think the whole thing would really change overnight if we had an unlimited supply of organs in terms of productivity of people post-transplant and the challenges that they face that are really exacerbated by the wait time and the fact that we don't have an organ available when someone develops end-stage renal disease.
The dream, the panacea would be that we could take care of the existing wait list with all the available options right now. Xeno, bioartificial kidneys, if people still want to be living donors, existing deceased donors, just take care of the entire list so we don't have to think about transplant, then go back to our own 2 beautiful native kidneys and use the entire armamentarium of drugs that we have now so we can preserve anybody who has CKD from not going to ESRD, and then move that needle up to maybe even reverse disease and one day never have kidney disease. That
Right.
would be the ultimate panacea. So
Well, that's the plan.
That's the
And then, really also to become inclusive of the people who never make it onto the list. So
Yes.
only about 10%, if you choose any organ, of the people who develop organ failure ever get listed for a transplant. And only a third of them actually receive a transplant. So we're transplanting about 3% of the people with organ failure every year. There's nothing else in medicine like that it is we know is the best approach, and we just have to ration it so much that it's really terrible. So that 90% of the people who end up dying from their organ failure, we want to begin to reach into that segment as well, with other sources of organs like xenotransplantation. And I think it also extends to lifespan and health span. If most people die from a single organ failing, and if we could more easily, have a replacement organ available to them, people who would add years to people's lives and their good health if we could do it at an early stage. So all of these things I think are our dreams, but our dreams I think are upon us because the rate of acceleration of everything now in science is extraordinary. And I think we're going to see things in 5 years that we imagine might happen in 30 years.
Bob, could I ask you to speculate about young infants. Do you think Xeno will have the capacity to push down the limit, the lower limit at which people can be transplanted?
Did you say the young people?
Infants.
Infants, yeah. So there's, I mean, perhaps the best case in transplantation right now or in organ failure are the children who have congenital heart disease, who need to get a transplant, a single ventricle. There are quite a few different congenital diseases that will lead to heart failure in the first year of life, or we may be able to do a surgical intervention that just pushes the need for the transplant down the road a bit. But we have this situation now where there's very few infant hearts available, very few donors, and so we're having to put these babies on what's called Berlin heart, which is a ventricular assist device. And believe it or not, we had this period of about 6 or 7 months where you couldn't get Berlin hearts, and it's old technology. It's like 30 years old, and there was a wait list for the Berlin hearts, which are sort of a bridge to a transplant. And kids were dying, because they couldn't, get a Berlin heart in time. Now that's been mostly addressed, but it's I think there's no other device right now in that space that is going to be able to. It goes back to the economics of these kinds of disorders that maybe don't fall into the rare disease definition that allows pharma to make big investments in rare diseases, and this is so I think these children are a really good group where the equipoise really would favor something like xenotransplantation, because the mortality rate is so high and the morbidity rate, 30 to 40% of these kids have strokes, on an assist device or while they're waiting for a transplant. And so that is, I think, a group where there's tremendous benefit from having a xenotransplant, either as destination therapy or a bridge to a transplant to take the place of these devices that aren't available.
So one last thing that we probably should mention is we all ask for well-designed, high-impact clinical trials. And if we are asking for that, then we've had a very well-designed, optimized study that was presented at Kidney Week 2 days ago. The older kidney patients, less likely to reject their organs, but more vulnerable to medication side effects. And so the optimized study tested whether less intense immunosuppression could improve their outcomes. So 379 participants randomized to compare everolimus and reduced dose tacrolimus versus mycophenolate and standard dose tacrolimus immunosuppression. And the lighter treatment regimen was safe, which is great. Did not show better survival or kidney function at 2 years after transplantation, despite the separation in curves with the tacrolimus levels. And so, beautifully done study. Did what it was supposed to do in terms of trial design and randomization, and didn't necessarily show that it was the tacrolimus levels, at least, that correlated with.
Yes. Yeah, I thought that was interesting because in a way it was a positive study for both arms.
Yes.
The one part of trial design is picking the population who's at risk enough to show that something works. If you're not going to get an event, it doesn't matter how good a drug is, you're not going to be able to demonstrate that in the trial. And the survival, both for graft and patient, in that older population over 65s was really, really good. I was staggered. That was a study done in Amsterdam, I believe. In
In Netherlands,
the Netherlands.
Groningen, Netherlands, yeah.
So I don't know if their outcomes are always good, but they certainly were for those
good,
participants. Of course, participants in trials always have better outcomes.
Yes.
There's a strong selection pressure there. One of my fellows did a study looking at all patients randomised into hemodialysis trials, and they have half the mortality of the background population. And as we know, there have not been many positive hemodialysis trials. So it just shows what selection pressure there is. To me, that comes back to your earlier point, Bob, about having platforms. The easier it is for people to be in trials, the more we're going to get the representative population that the evidence is ultimately going to be applied to. So number one, we'll get patients who are at risk, so that will accelerate successful trials. But number two, we'll then be much better placed to generalise the results of those patients.
Can you give us an example of some of the work that you're doing that would kind of fit into that?
I'm glad we're
I think you were talking about
2 hours. I've got a lot to say.
calcinosis trial.
Well, now one example maybe that's particularly relevant to trials in rare disease is our Beat Calci trial, which is a platform trial, and it's looking at ways to improve outcomes for calciphylaxis. There's, a rare but devastating ulcer-associated condition in people, generally in people receiving the dialysis treatments. And, survivors vary, but in some reports the median survival is 3 months, and it's a very painful condition. Because it's so rare, though, it's been very hard to conduct trials, and industry has tried, and academics have tried, and we have very few completed trials. So we thought the way to address it was to have a platform, involve as many countries as we can. If anyone's listening, please contact me. And to use the efficiencies of some of the adaptive trials design embedded in that platform. So in one domain, we're looking at repurposing medications which are currently used spasmodically with no evidence, and we've got a shared control. So you instantly get efficiency there. We have our endpoints to determine when you get to the standard of proof rather than testing on a sample size. So what that means is if something's strongly effective, it's going to hit that mark earlier and graduate earlier. If something's not effective or weakly effective, it will take longer. But that's what you want, isn't it? You want the most effective things to come through earlier. And the other thing about it too is we're designed it so we have a domain for every aspect of care. So these patients all have some form of dialysis, they have systemic therapies, they have topical therapies. We'd love to have a novel agent domain as well, so that patients can go in as many of these at the same time as they like, and that will really hasten the generation of evidence.
Yeah, I mean, the openness of the FDA to these adaptive type of trial methodologies, and also the trial that we're doing now, Expand, which is looking at a 10 gene edited kidney, and it is designed so that we have a safety run in, so kind of replacing a phase 1, phase 2 type of trial, six of those transplants, and then based on the data, there'll be a committee that will review all of that and decide on endpoints at that point that make the most sense, and then it'll seamlessly then go into a phase 3 trial, and that those kinds of trial,
Efficiencies.
Yeah, exactly. It makes such a difference in the time to getting an answer, and the likelihood of getting an answer that is easier to interpret. I mean, I think it can really speed things along, and also I think it helps to keep pharma engaged. When the whole trial is basically constructed, from the start, like you can see the end of that and plan for it, much easier.
There's a map.
Yeah.
And I think one of the biggest advocates for this are the patients and the patient organisations. They don't want to wait. It's personal, and so they are really strong on pushing for efficiencies. And I find often having patients in the room, they're more progressive on this than many clinicians, because they know the importance of progressing the evidence. We're doing another group that's very underserved is the polycystic kidney disease community. Although that is starting to change, there are so many drugs in early phase development, and so groups of us around the world are trying to put together a platform there to develop that vehicle for really pushing agents through the trials. So it's exciting. It's not easy, but it's exciting, and the patients are making things much more possible.
The willingness of them to participate in trials. And
Whatever
them
the
being-
barrier is, sometimes it might be regulatory, sometimes it's ethics. Patients have a lot of clarity on what's a sensible thing to do, and they're very persuasive.
Yeah, and I think, patients, I'm a patient, I've had a heart transplant, and look, there's no one who really understands the stakes better than a patient who's, waiting for an organ. And I think that, patients are much more willing to take risks than physicians and the people running these trials and the pharmaceutical companies. And they're now really beginning to demand that this, that we start to get answers more quickly, and that the trials are designed in ways that make sense, that don't, lead to very little hope in your lifetime of, getting a new drug or having whatever condition you have solved and cured. And I think we need to listen to the patients.
That point you're making there about the background risk, that is so important. There's a totally different setting if you already have kidney organ failure and looking at potential things that will improve your outcome. Then it's quite a different scenario. You might be willing to take a whole lot more risk than if you're a basically well patient and there's a preventative agent. So, being really thoughtful about what the risks are of not having a trial, I think that's an important component of all of this.
The willingness and the openness to have patients as part of scientific advisories, clinical trials, FDA panels, having a breadth of patient experiences, and that has been another really exciting, necessary change in the recent years, and is very evident in Kidney Week. We have a lot of patient representatives.
Yes. In fact, I would submit that, the reason why things are happening so rapidly in xenotransplantation and the openness that the FDA has to these trials now is because of a meeting that was brokered by the National Kidney Foundation to bring patients together with the FDA to talk about trials in xenotransplantation. And there's no question that we saw a really different approach from the FDA after that meeting occurred. It had a dramatic impact. And I think we need, no one really understands, as clinicians, as physicians and surgeons, we're just seeing the very tip of the iceberg of what the impact of a disease has on patients. We don't know what it's like to walk in their shoes, really, unless we do at some point. I mean, we all will eventually become a patient, but so I think, these interactions are really very, very important.
I think that's a beautiful note to end this on. Yeah, that we're here because of our patients. We have an identity because of our patients, and maybe continue to serve them. You already do, but we hope to get there. What a pleasure this has been to share this
thank you all.
table
Yes,
with
thank you.
you.
Yeah, it was a
fun.
good conversation. And it's been a great conference, hasn't it? I'm looking forward to next year's.